For centuries, smallpox was one of the most destructive forces on Earth. The virus moved through human populations with terrifying efficiency, collapsing empires and killing up to 60% of the people it infected. Those who survived were often left blind or covered in deep, disfiguring scars. Humanity's first deliberate countermeasure was a crude procedure known as variolation. A practitioner would take a lancet, slice into the skin of a healthy person, and deliberately rub in the live pus from a smallpox patient's blister. The goal was to trigger a mild infection that would grant the patient lifelong immunity. This bar chart compares the mortality rates during the 1721 smallpox outbreak in Boston. The data shows that naturally contracted smallpox killed roughly 14% of the city's infected residents. For those who underwent variolation, the mortality rate dropped to just 2%. But the procedure was entirely unpredictable. Patients could develop severe, fatal cases of the disease, or unknowingly spark entirely new epidemics while they recovered. Early medicine was effectively playing Russian roulette with a pathogen, trading a massive risk to the individual for a statistical advantage to the population. Curious, Tone? In 18th century Europe, a specific rumor circulated among farming communities. Young dairymaids who caught cowpox, a mild infection passed from the udders of cattle, seemed to be completely immune to the ravages of smallpox. A few rural pioneers decided to test this folklore themselves. In 1774, a Dorset farmer named Benjamin Jesty took a darning needle, scraped pus from an infected cow, and intentionally scratched it into the arms of his wife and 2 young sons. The family remained immune to smallpox for the rest of their lives. Yet the discoveries made by Jesty and other rural practitioners like apothecary John Fuster barely registered in the broader medical community. They performed their inoculations in isolation. They collected no systematic data, published no empirical findings, and made no effort to promote their results to the scientific establishment. A correct observation is only the first step in scientific progress. To convince the world to adopt a radical new medical procedure, you need undeniable, systematized proof. That proof arrived through an English physician named Edward Jenner. In May 1796, Jenner decided to formally test the cowpox hypothesis. He extracted material from a dairymaid's fresh cowpox lesions and inoculated a healthy 8-year-old boy named James Phipps. After the boy recovered from the mild cowpox fever, Jenner took a terrifying next step. He intentionally exposed the child to live, lethal smallpox material. When the boy developed no symptoms, Jenner concluded that the protection was absolute. By today's standards, Jenner's methodology is alarming. Exposing a child to a lethal virus would instantly violate the Nuremberg Code. The Nuremberg Code is a set of ethical research principles for human experimentation, established after World War II to protect the rights of research subjects. This is a psychological phenomenon where vulnerable parents fail to distinguish between clinical research and proven treatment, mistakenly believing an experiment is designed specifically to heal their child. Under extreme stress, they cannot provide truly objective, informed consent. We are left with a profound historical paradox. The foundation of immunology was built on a pediatric experiment that would be universally condemned as a severe ethical violation today. In 1798, Jenner privately published his findings in a booklet outlining his experiments. By documenting the exact methodology and tracking the outcomes of multiple patients, he transformed the physical act of vaccination into a replicable, data-backed scientific protocol. As Jenner's data crossed borders, the medical establishment responded. Governments prohibited the highly dangerous practice of variolation, and widespread vaccination campaigns took its place. That protocol set a global eradication process in motion. Following an intensive, synchronized campaign by the World Health Organization, the medical community reached the ultimate conclusion of Jenner's work. In 1980, the WHO officially declared that smallpox had been wiped from the face of the earth. The eradication of smallpox proved that when empirical data is systematized and deployed on a global scale, humans possess the capacity to permanently delete nature's most entrenched killers. Modern immunology has moved far beyond scraping animal blisters. Today, biotechnology operates at the digital and molecular level. Researchers now use an approach called reverse vaccinology 2.0. This methodology involves decoding the human immune system at a single cell level, scanning blood from recovering patients to identify highly potent antibodies. These antibodies are identified by analyzing B cells, which are white blood cells that produce the specific proteins needed to neutralize pathogens. By isolating these exact antibodies, scientists can reverse engineer the pathogen structure. For instance, researchers analyzed human antibodies to map RSV, a common virus that can cause serious lung infections. They then designed a stabilized pre-fusion protein, a version of the virus locked into its harmless shape before it ever enters a human cell, and used it to train the immune system safely. Scientists are taking this even further with germline targeting peptides, such as the EOD-GT8 60mer, designed for HIV. The EOD-GT8 60mer is an engineered nanoparticle that activates the specific precursors needed to produce HIV-neutralizing antibodies. This allows researchers to shepherd the body's precursor cells, coaxing them to produce broadly neutralizing antibodies before a virus ever attacks. Modern vaccinology now operates at a design level, allowing us to program the specific architecture of the body's defenses before a threat ever appears. This molecular precision is essential because we are facing a biological crisis, antimicrobial resistance, or AMR. AMR occurs when bacteria and viruses evolve to survive the drugs, such as antibiotics, originally designed to kill them. This graph forecasts the deadly trajectory of that resistance. Without new interventions, the death toll from antibiotic-resistant bacteria is projected to grow, reaching 10 million people annually by the year 2050. But as this chart illustrates, vaccines possess a distinct tactical superiority. While pathogens can easily mutate around antibiotics that only attack a few specific metabolic targets, vaccines train the immune system to launch a broad, multi-target defense. Pathogens rarely evolve resistance to that level of biological pressure. The core principle behind Jenner's cowpox experiment remains our most effective strategy. While pathogens adapt to our chemical treatments, a programmed immune response provides a stable defense for a world where antibiotics may no longer work. In September 1854, the Golden Square neighborhood of London became a graveyard overnight. People who woke up healthy were collapsing in the streets and dying in pain before the sun went down. The mortality rate was staggering. In just a few square blocks, 500 people were killed in just 10 days. Hospitals were paralyzed by the influx. Physicians tried bloodletting, opium, and castor oil, but the disease continued to tear through the population, causing massive dehydration and organ failure in hours. Authorities were helpless. They were losing a war against an enemy they didn't understand, and their existing medical knowledge offered no way to slow the spread. Most scientists at the time believed in miasma, the theory that diseases spread through bad air and the foul smells of rotting organic matter. Since London's industrial streets were notoriously pungent, the air itself was blamed for the deaths. Dr. John Snow disagreed. He had spent years investigating cholera and suspected it wasn't something people breathed, but something they swallowed. He believed the source was contaminated water. Snow couldn't simply argue against the medical consensus with a hypothesis. To convince a skeptical establishment, he needed a way to make the evidence visible and mathematically certain. He set out to anchor the disease to physical geography, turning the street map into a tool for tracking a killer. This is Snow's grand experiment. He tracked cholera mortality across South London, where two water companies had pipes running down the same streets. One company took water from a sewage-polluted Thames, the other had moved its intake upstream to cleaner water. But his large-scale study was interrupted by a sudden, hyper-localized outbreak in Golden Square. Snow dropped everything and rushed to the Soho crisis. He began a grueling door-to-door investigation, interviewing families about the exact water habits of those who had fallen ill. To ensure his count was accurate, he collected the precise addresses of the first 89 deaths from the General Register Office. By combining official records with street-level interviews, Snow created a dataset linking the biology of the disease to the geography of the neighborhood. This is the result. Snow plotted each recorded death at its specific location on a map of Soho. Each black bar represents one person killed. The visualization revealed a massive cluster around one specific point, a public well on Broad Street. To reinforce the point, Snow added a mathematical boundary called a Voronoi diagram. It showed that for almost every victim, the Broad Street pump was the closest source of water by walking distance. While Snow mapped the area, Reverend Henry Whitehead conducted his own interviews. Initially a skeptic, Whitehead eventually found the index case, the first infant to fall ill at 4 Broad Street. Whitehead discovered that the infant's soiled washwater had been dumped into a decaying cesspool. That cesspool was leaking directly into the well of the Broad Street pump. Snow's map identified the target, and Whitehead's forensics found the source. Together, they had built an airtight case for waterborne transmission. On September 7th, Snow presented his evidence to the local Board of Guardians. The very next day, they removed the handle from the Broad Street pump. With the primary source of the pathogen cut off, the outbreak came to a halt. Despite the results, the General Board of Health officially rejected Snow's report. They remained committed to the theory of bad air and dismissed the map as a coincidence. They weren't the only ones mapping the data. An engineer named Edmund Cooper had mapped the deaths for a sewer commission earlier, but because he was trying to prove that sewer gases were the cause, he failed to see the pump at the center of the cluster. It proved that even with the correct data on the page, the establishment remained blind because they were using the wrong theoretical framework to interpret it. Snow's argument was so mathematically sound that it eventually broke the consensus. His work led to a complete overhaul of London's sewer and water systems, finally separating drinking water from waste. This single revolutionary map didn't just stop a local outbreak, it helped define the modern fields of epidemiology and data science. The process of layering infection data over a physical grid remains our primary method for tracking and containing infectious diseases today. Removing the pump handle stopped one localized outbreak, but the map that led Snow there established the protocol we still use to stop invisible killers. In 1921, a diagnosis of type 1 diabetes was an absolute 100% fatal death sentence. At the time, the only medical intervention was an extreme starvation diet designed to merely delay the inevitable. These restricted calories took a horrific physical toll. Patients like Leonard Thompson weighed only 65 pounds, and Elizabeth Evans Hughes dropped to a skeletal 45 pounds. Children wasted away in hospital wards like these, living in a grim atmosphere while constantly hovering on the brink of a diabetic coma. Before 1922, the entire global medical establishment was completely powerless to stop this metabolic countdown. If you know the story of insulin, you likely know the legend. A young, inexperienced Canadian surgeon named Frederick Banting wakes up with a late-night idea that will lead to a cure. The popular history says that Banting and a young student, Charles Best, isolated insulin from dogs, single-handedly cured the disease, and became international heroes. While that lone genius story is famous, it entirely erases a massive scientific bottleneck, a near-fatal first trial, and the 2 other scientists who actually made the drug safe for human blood. The core biological problem in the 1920s was that while the pancreas secretes an anti-diabetic hormone, it also produces a digestive enzyme called trypsin. Trypsin destroys the hormone during extraction. Banting's surgical workaround was to tie off the pancreatic ducts in dogs, killing off those trypsin-producing cells and leaving the precious anti-diabetic secretion intact and unharmed. To test this theory, leading diabetes expert J.J.R. McLeod provided Banting and Best with the crucial laboratory space, test subjects, and academic guidance they needed. The initial results were promising. Banting and Best produced a crude extract that successfully lowered blood sugar in dogs, but the liquid was a toxic, thick brown muck loaded with dangerous impurities. On January 11, 1922, this crude extract was injected into 14-year-old Leonard Thompson. It failed, causing a severe allergic abscess instead of a cure. Banting and Best had hit a scientific wall. They had proven the biological concept, but they lacked the biochemical skills to refine that animal extract into a usable medicine. To solve the impurity problem, McLeod brought in J.B. Collop, a brilliant biochemist. Collop used a systematic biochemical method. He dissolved the crude pancreatic tissue in a solution and began gradually increasing the concentration of ethyl alcohol to strip away layers of impurities. When Collop pushed the alcohol concentration past 90%, the final impurities were trapped at the top, and the pure active principle, insulin, precipitated out at the bottom. On January 23rd, this purified extract was given to Leonard Thompson in a second human trial. This time, his blood sugar normalized, and his starvation symptoms vanished. It was Collip's chemical purification protocol, not Banting's initial surgical idea, that was the actual catalyst that turned a toxic animal extract into a life-saving human medicine. Immediately following the discovery, the team fractured. Banting felt intense insecurity over Collip's chemical breakthrough and resented MacLeod's oversight of the project. The team did achieve one unified ethical victory. They secured the patents for the production method, but sold them to the University of Toronto for $1 each, ensuring no single company could monopolize the cure. However, a bitter public relations war followed. Banting and Best launched an aggressive media campaign to claim sole credit, painting MacLeod as an idea thief and almost completely erasing Collip's contributions. The 1923 Nobel Prize Committee attempted to recognize the collaborative reality by awarding the prize jointly to Banting and MacLeod. The fallout remained bitter. Banting split his prize money with Best out of spite, while MacLeod split his with Collip. Ultimately, the history books adopted Banting's sanitized version of the story. That aggressive media campaign successfully airbrushed MacLeod and Collip from popular history, rewarding the men who had the initial idea while erasing the men who actually executed it. Fast forward to today, and that messy, fractured discovery has laid the biological groundwork for a century of complex, award-winning science. Later researchers spent decades demystifying the drug, such as Frederick Sanger, who successfully mapped insulin's amino acid structure. An amino acid structure is the specific sequence of chemical building blocks that make up a protein. Rosalind Yalow later developed radioimmunoassays, a technique that finally allowed doctors to track exactly how insulin metabolizes in the human bloodstream. A radioimmunoassay is a highly sensitive laboratory technique that uses radioactive isotopes to measure the exact concentration of specific substances in the blood. These breakthroughs are almost never the result of a solitary researcher having a sudden realization. Instead, they are forged through iterative, messy, and deeply collaborative human effort. In the 1920s, a simple bacterial infection often meant a slow and inevitable decline. Hospital wards were crowded with patients dying from common pneumonia or infected wounds, as physicians had no effective way to stop the spread of bacteria. The popular narrative of how this changed begins with Alexander Fleming returning from holiday in 1928. He discovered a messy lab, an open window, and a stray speck of mold that had accidentally landed in a petri dish and killed the Staphylococcus bacteria around it. This sequence of events is often framed as a stroke of pure luck, a fortunate contamination that saved the modern world. But attributing global breakthroughs to random chance is a simplification. It ignores the specific psychological traits required to turn an anomaly into a discovery. To find the actual drivers of innovation, we can look at a massive library study from the 1980s. When hundreds of universities were forced to reshuffle their journals due to a new cataloging standard, it created a natural experiment in how scientists encounter unexpected information. This chart tracks the innovativeness of the resulting scientific papers. Simply being exposed to random, unsought information didn't change much. The baseline for citations remained almost perfectly flat. The data only shifts when we isolate a trait called openness. In science, openness is the ability to recognize and extract value from data that contradicts your initial goal. For researchers with this trait, the library reshuffle led to a measurable increase in innovative citations. In contrast, scientists with deep experience but low openness actually cited the new journals less. They relied on what they already knew, effectively ignoring the new information they bumped into on the shelves. The penicillin discovery wasn't a product of an open window, but a mind ready to investigate an error. In 1928, Fleming wasn't just waiting for a fluke. He was systematically hunting for antibacterial agents. He had been studying lysozyme, an antimicrobial enzyme found in human tears and egg whites that can dissolve the cell walls of certain bacteria. When the penicillium mold contaminated his Staphylococcus plate, he didn't see a ruined experiment. He saw an anomaly that matched his ongoing search for bacterial killers, and he pivoted his entire focus to investigate why the bacteria were undergoing lysis. His laboratory notebooks from the 1930s prove he didn't abandon the project. He spent a decade testing other airborne molds and quietly documenting how they interacted with various pathogens. Fleming's openness provided the first observation, but turning a microscopic observation into a mass-produced drug required a massive industrial coordination. By World War II, the need was urgent.
Allied medics needed a stable, mass-produced antibiotic to treat the thousands of casualties suffering from wound infections on the front lines. Scientists Howard Florey and Ernst Chain at Oxford proved that penicillin worked in humans, but they couldn't produce enough of it. To scale the drug, they had to move the search to the United States. This search for a hardier, high-yielding strain ended at a local market in Peoria, Illinois. A laboratory technician named Mary Hunt found a moldy cantaloupe that hosted a rare mutation, a strain that produced over 200 times more penicillin than Fleming's original mold. USDA scientists then developed submerged deep tank fermentation. By growing the mold inside massive, aerated tanks of corn steep liquor rather than on flat plates, they could harvest the antibiotic on an industrial scale. Determining the precise structure of the penicillin molecule fell to Dorothy Hodgkin. She used X-ray crystallography, a technique that uses X-ray beams to map the arrangement of atoms in a crystal, to prove the drug's architectural design. This coordination between microbiology, industrial engineering, and chemistry turned a laboratory accident into an industrial reality. The resulting golden age of antibiotics saw a 40% drop in infant mortality and a significant increase in general life expectancy. Diseases that were once fatal became routine infections that could be cleared in days. This era demonstrates that significant breakthroughs are not passive gifts of luck, but outcomes of minds trained to investigate the unexpected. In 1945, as he accepted his Nobel Prize, Alexander Fleming offered a prophetic warning about the future of his discovery. He warned of antimicrobial resistance. This is a condition where bacteria evolve to survive exposure to the drugs designed to kill them, often because the drugs are used in doses too small to eliminate the entire infection. To survive the coming post-antibiotic era, we cannot wait for a window to blow open. We must rely on the same rigorous openness that built the original breakthrough.